Let me tell you a little bit about Dr. April Pyle, who is the recipient of your endowed chair. April obtained her bachelor's and PhD degrees from the University of Tennessee in Knoxville. So I guess that makes her a country girl. She then joined Peter Donovan's research group for a postdoctoral fellowship. And that was initially at Thomas Jefferson University in Philadelphia. And subsequently, they moved to Johns Hopkins in Baltimore. And it was there in Dr. Donovan's lab that she first became interested in human embryonic stem cells and in pluripotent progenitor cells. We hired April here to UCLA in collaboration with the Broad Stem Cell Research Center in 2006. She's now a full professor, and she's also a vice chair of our department, and her responsibility is to oversee undergraduate education in our department. Her recent work is focused on the use of pluripotent stem cells in the study of muscular disorders, and as George said, in particular, Duchenne muscular dystrophy, which is also known as DMD. She does this both in terms of modeling the disease to figure out exactly what's happening, what's the pathogenesis, and also in developing novel therapies. So DMD is a debilitating muscular dystrophy that generally affects young boys. It's caused by mutations in the dystrophin gene. Remarkably, April's research group has been able to devise a molecular strategy to correct these mutations, and she'll tell you more about that in detail, hopefully in a few minutes. She's shown that this system works in tissue culture, so in the lab, as well as in animal models, which I think is the most exciting component. Being able to deliver a therapy like this in vivo is really spectacular. Importantly, April is interested in translating these results to the clinic. And as you heard earlier, part of this endowed chair award was for someone with entrepreneurial spirit. So April has co-founded a company, as George mentioned, called Myogene, and she would like to bring these therapeutics to the clinic. And hopefully with her work and that of others, a cure for diseases such as DMD will arise shortly. So on the more personal side, April is highly enthusiastic, probably one of our most enthusiastic faculty members. She's highly sought after as a collaborator, and she's an outstanding colleague. She takes incredible pride in passing her knowledge on to her trainees, her students, her graduate students, and her undergrads. She also takes great pride in helping the department and the campus. In short, she's the perfect inaugural recipient for this chair. So with that, I would like to ask April to come up to the mic and showcase your work. And April, congratulations.
With that, let me just kind of tell you what we live and breathe about every day, and that is stem cell biology. And so, as Jerry said, I came here with an interest in stem cells, and particularly what it is that makes these stem cells unique. What are the cells in our body that have the ability to both make more of themselves? This is a process called self-renewal, as well as differentiate into any cell type in the body, especially if we're talking about human pluripotent stem cells, as you mentioned. And so one of the important control features of this is that stem cells are tightly balanced in how they divide or differentiate. And if this process goes awry, then this could lead to abnormalities in terms of cancer or in disease settings. And so understanding the basic machinery that controls stem cell function, how a stem cell makes another copy of itself versus go down a path for making a special cell type in a tissue, a specialized cell type in a tissue, is it's a basic question, but the implications of that are huge. And so by being able to study that, which I've been doing at UCLA since I started in 2006, has been remarkable. And one of the things that excite me the most really are the power of human pluripotent stem cells. So there's two types of stem cells in the body. There's what are called pluripotent, or there are adult stem cells, which can be multipotent or unipotent. And so pluripotent stem cells have the ability to differentiate into any cell type in the body. Most adult stem cells are multipotent, such as the hematopoietic stem cell, which can give rise to multiple blood cells of the blood lineage, or in the case of what we study, muscle, which typically gives rise to more myocytes in muscle in a dish. So we study the properties of these cells, both pluripotent and adult, to try and develop strategies to both understand basic research and clinical applications. And so this highlights that from pluripotent stem cells, we can use these as a tool to understand human development. And that's something that really is the foundation of my lab, is understanding how these cells specify in a dish with the hope of making better products for disease discovery. We also use pluripotent stem cells in drug discovery. So having a patient-specific cell that you can make with pluripotent stem cells, you have the genetic makeup of that patient. So then you can tailor therapeutics or drug discovery in that context of personalized medicine. And then finally, one of our long-term goals is to think about using pluripotent stem cell-derived cells in cell-based therapies. So as I mentioned, we study a type of adult stem cell called the satellite cell. And contrary to what you may hear, not all organ systems have endogenous stem cell, but skeletal muscle does. And the skeletal muscle stem cell has a remarkable property. This cell has the ability to make more muscle upon injury, and it's due to a very rare cell in muscle called the satellite cell. And this cell actually sits in a unique location, as shown here. And if you take a cross section of this muscle shown to the left, you can see that the muscle is beautifully brimmed with this protein called dystrophin. And these compact structures that are rimmed with dystrophin are the individual muscle myofibers. In looking below that, you can see the blue dots. About one of those blue dots is going to be the stem cell. So it's a very rare cell. And that cell is remarkable because upon injury can give rise to whole new myofibers. And so those properties of stem cell biology are what we're really excited about, and we look to harness in our lab, both studying both the basic questions of stem cell biology and therapeutics, as I said. And the way that we've really embarked on this has been in large part through using human pluripotent stem cells. So we're very lucky to be at UCLA because it's a hub of stem cell biologists, and a lot of the initial reprogramming has been done by many members of the Broad Stem Cell Center, and this has been instrumental in our success in our lab. We've also been lucky to be a part of a muscle center that has enabled me to really learn about Duchenne muscular dystrophy, skeletal muscle, and really think about approaches that might be relevant in the context of skeletal muscle diseases. And so in our lab, we have developed Duchenne muscular dystrophy disease models in a dish. And so this is just showing you kind of the circle of what we think about in the lab. And so what we've done is taken patients in the clinic that have DMD, and DMD is due to mutations in the largest gene in the genome called dystrophin, and I'll show you that in the next slide. We've taken these patient cells back in the lab and reprogrammed these using overexpression of key pluripotency genes, and then we've made these human-induced pluripotent stem cells. The challenge is that now that we've made these human-induced pluripotent stem cells, they still hold the mutation of the patients, right? So they still have mutations in dystrophin. And so to think about delivering back cells that are affected in these patients, mainly these patients are affected in the heart and in the skeletal muscle. So we would want to restore back skeletal muscle or cardiomyocytes to these patients. The challenge is that you still have the patient mutation in a dish. And so we actually have one of the most talented grad students who's here on the call, Courtney Young, who actually in the lab in collaboration with Melissa Spencer developed a CRISPR-Cas9 gene editing strategy to try and fix these mutations. And so I'll tell you a little bit about that work. So we've really spent a lot of time trying to think about strategies to fix the mutations, if you will, in the context of a human pluripotent stem cell model. And we've also spent a lot of time in the lab trying to understand how muscle is developed in a dish from human pluripotent stem cells, because if we can make a stem cell in a dish, then we have the power of that cell to regenerate in when we can give that back, hopefully eventually to patients, because this cell has the ability to continuously make new muscle anytime there's an injury. So what I want to tell you about just briefly is our interest in generating skeletal muscle in a dish, our strategies for using CRISPR and gene editing strategies for muscle and stem cells, and then new work that we're thinking about developing disease models for other neuromuscular diseases. So one thing that you may not realize is that because we're working with pluripotent stem cells, these cells are very early in development. And so it's almost like a clean slate. And so one of the biggest challenges in the lab has been to try and understand how to guide these cells. These cells are very plastic, and if you don't watch them, they're like children, they'll go in a million different directions. And so we have spent a lot of time trying to understand the cues that will teach these cells how to make specific mesoderm subtypes such as skeletal muscle. And we've also been interested in understanding how human development does this, because we want to use this as a roadmap to understand when we have the right cell, where does it compare to with the cells that we make in the human body. And so we've been trying to understand the differences in what we call skeletal muscle progenitor cells, which arise early in human development, versus the cell that's sitting on the outside of the myofibers and muscle, the adult stem cell. And to do this, we've used human pluripotent stem cells as a way to understand how to direct these cells to what I call the gold standard muscle stem cell. So we embarked on a crazy project. I remember sitting at the blackboard in my office, and this was just when a platform had been developed at Harvard called drop sequencing. And this was a way to flow single cells through a fluidics device and capture gene expression in individual cells. And I said to my postdocs in the lab, I want to do this for all of human limb development. And they said, you're crazy. We'll never be able to get the tissues. We'll never be able to do this. We're going to get scooped. It's going to take time. And I said, well, we have to do it because we have to know how these muscle cells develop so that we can make them in a dish. It's key. It's like step one. And so we embarked on this journey. It was at least a 3-year journey of collecting tissue from every source we could, as well as developing strategies to differentiate these cells in a dish. And so we collaborated with Catherine Plath, who built a drop-seq sequencing platform in her lab, and we used this approach to look at genes that are expressed in single cells throughout human development. And we did this across embryonic, fetal, juvenile, and adult skeletal muscle. And there's going to be a quiz on this later, so don't worry. But no, really, this is just a snapshot of some of the data that we've collected over time. And I don't expect you to remember all these different cell populations, but the point is to highlight that development is dynamic, and the cells that we make in a dish are also trying to recapitulate that. And so one of the things that this is showing here is the circles are us identifying the muscle cells that are present in each of these stages. And then the other cells that are there are all of these other colors, are actually other cell types that are present in the human limb. And so this is important because, as shown here, the cells that are surrounding the stem cells are what crosstalk to that cell in development. And you can see here, just by looking at the color changes, that we have dynamic transitions in human development of these supportive cell populations. And so these are providing cues as to tell the stem cell how to specify and develop over time. And so we're trying to harness this information to be able to develop better muscle stem cells in a dish. And this also kind of summarizes some of the recent work we did that was carried out in large part by Hyben in collaboration with the Plath Lab, where we took all of these different samples that are sort of shown here, and we also compared across multiple protocols to try and identify the developmental trajectory of these cells. And this circled population here in the middle, called skeletal muscle progenitor cells, show that the cells that we make in a dish from human pluripotent stem cells align with about human week 7 to 12 in development. And really all of this work was to try and identify this staging so that we have the right that we're at the right starting point. We know what we're making, and now we know where to go from here because we know what the adult satellite cells express. And so now what we want to do and have been doing is using this information as a tool to guide the ability to make a more mature stem cell in a dish from a human pluripotent stem cell. And this just shows you our efforts on being able to do that. So in this beautiful directed differentiation from a human pluripotent stem cell in a dish, we've been able to make skeletal muscle. And this skeletal muscle is unique in that it's a multinucleated muscle cell. And so it has multiple nuclei. I don't know if you can appreciate this, but sometimes you can see hundreds of nuclei in one single muscle fiber. And this is something that we're now able to begin to recapitulate in a dish from human pluripotent stem cells. This is work done by Michael, who's on the call. And this is also showing work where Michael has spent many years trying to develop a model of how we can look at the functionality of these cells. So we know their gene expression, we know what genes they turn on and off, but how truly functional are they? So if you put these cells in a model of disease, they start to turn on proteins that are these green fibers that you see here. The human cells are in red, and they really begin to spread in regions throughout the muscle where you put them. And so we're making a lot of progress in developing these models in a dish to be able to understand the true functional potential of the cells that we make. So why is this important? Why are we trying to do this? Well, as I said, a lot of times the stem cell, the muscle stem cell, is the organ, is the cell of repair in muscle. And so this balance is normal in most of us, right? And so we have the balance of telling this cell to make more, to expand, to make progenitors, to make new muscle. But a lot of times in neuromuscular diseases such as DMD or in aging or in other neuromuscular diseases, you see a misbalance of this. Sometimes you have lack of progenitor cells that can be made. Sometimes you have disrupted self-renewal, and so, or progenitors that are produced. And so what we want to do is to be able to replenish these populations, if possible, that are abnormal. And the disease that we've really embarked on thinking about the most is Duchenne muscular dystrophy. And so as you heard, Duchenne muscular dystrophy is a devastating skeletal muscle wasting disease. It's due to mutations in the largest gene in the genome. And this leads to premature death. It's X-linked, so it's present in about 1 in 5,000 boys, and there is no cure. So these boys become wheelchair bound in their teens, and they die in their 20s. And so and they typically die of heart and respiratory muscle failure. And so a number of groups are trying to think about therapeutic strategies to really extend the lifespan of these boys. And so when Courtney joined the lab, as I mentioned, in collaboration with Melissa Spencer, we started thinking about another disease that's called Becker muscular dystrophy. And Becker muscular dystrophy is shown to the right here. And if you compare that to first of all, on the left is the wild type, and you can see that the dystrophin is this long protein that spans the muscle sarcolemma into the cell. And this really acts as a shock absorber because remember, muscle is a contracting cell. And so in DMD, there's no dystrophin protein that's made. In Becker, which is an allelic disease, there is dystrophin protein made. However, it's just truncated. So where this box is, you can see that these patients actually make a truncated version, and they're able to kind of restore this complex that's sitting on the top of the muscle membrane, whereas in DMD, this complex is missing. And that's shown at the bottom, where you can see this beautiful rimming of the myofibers and green of dystrophin. In DMD, you have no dystrophin. And then in Becker or BMD, you have some dystrophin, although it's not exactly the same as wild type. You're able to restore levels that would really improve functional gain in these patients. And so we wanted to try and sort of think outside of the box and develop a strategy to turn a DMD into a BMD. And the reason we wanted to do this also is because there is a hotspot of mutations within exons 45 to 55 of the DMD gene. And so we wanted to think, can we actually develop a gene editing strategy that could actually remove this entire region in the gene? And I know that sounds crazy. Why would you remove a region of the gene? But we actually wanted to do it so that we could then replicate a Becker-like protein in the dish. And this is a hotspot of mutation. So patients with Duchenne muscular dystrophy, at least 50% of these patients have mutations within this region. So instead of trying to tailor a strategy to each mutation, we thought, let's try and treat as many patients as possible with removing this entire mutation region, as you can see here in this graph. And so this is a beautiful cartoon that Courtney developed showing that in Duchenne, you actually have a mutation that's in the circled region where the puzzle pieces don't fit together, right? And this encompasses about 50% of mutations. We wanted to develop a strategy, which I'm sure many of you have heard now, CRISPR, Cas9 gene editing, where we can actually guide the cutting protein, which is Cas9, to these RNAs, which are called guide RNAs, which are the flags, and Cas9 is represented by the scissors to actually cut out this region. And when we did this, the hope was that we could fit these puzzle pieces back together and restore the dystrophin protein. So now the puzzle pieces would fit together if we've been able to restore dystrophin. And long story short, we were able to develop this platform. You can see by deleting this whole exons 45 to 55 region in the gene that we've been able to restore. And this is just shown here in cardiomyocytes in green on the left and on the right in skeletal muscle that we've made from human pluripotent stem cells. So we've been able to develop this strategy personalized with patient cells in a dish to be able to fix this mutation region and restore dystrophin in these cells in the lab. Also in vivo, we developed a humanized mouse model, so it has the human gene in the mouse model of Duchenne muscular dystrophy. And we packaged this CRISPR-Cas9 machinery in a viral system called AAV. And then if we inject this, as shown to the left, directly into the muscle, we get new dystrophin positive fibers or restoration of dystrophin as shown here in these red fibers. Also below, these bands show that we've restored dystrophin in these mouse models. And to the right, if we deliver this to every muscle, so we call it systemically, which would go to both the heart or the muscles, you can see that we've been able to restore dystrophin, rimming these individual myofibers in vivo in the animal models of the disease. And so surrounding this work, we've developed a lot of IP over this technology and many others. And I just have to highlight that this work was not done alone. This involves many collaborations, many faculty, many trainees, where we're kind of looking at what are the best strategies to restore this dystrophin gene? How do we deliver it? We've tried a number of different delivery strategies. AAV currently seems to be the most efficient. And in collaboration with Courtney and Melissa, we've been thinking about ways to dampen the immune response to be able to deliver this to patients. Michael Hicks, also in the lab, really has been spearheading how to generate skeletal muscle progenitor cells in a dish. He has a patent on this work. And so we're moving forward on both strategies, both cell-based and viral-mediated delivery, so that we can try and cure these patients. To do this, we really wanted to kind of step out of the box and think about how to merge our findings to get to the patients the quickest. And to do this, we developed a company, as you heard, called Myogene Bio. And this is actually housed in the CNSI Magnify incubator right now. It's founded by Courtney Young, who you've seen, who's here, who did all of this amazing work. She really is a superstar. She did this as a grad student. And she really is one to watch. And she's CEO, co-founder, and we developed this company, Melissa and I, and we're really in the process of pushing this forward. We're trying to raise a $25 million Series A to move this to the clinic as safely and effectively as possible to cure DMD patients. So that's a large plan moving forward. Safety is key. Delivery. How do we deliver this safely? is the best way AAV? Can we get stem cells to deliver this, to provide a corrected pool of stem cells? And so we're really working on both of those fronts, to both on the basic side to improve it, as well as moving it forward to patients. And the last direction that I'll tell you about, just the last 2 slides, is another important feature of skeletal muscle, which is the innervation by the motor neuron. And so in a lot of neuromuscular diseases, there's a dysfunction or a loss of connection of this innervation to the muscle in aging or other neuromuscular related diseases. You can see that the muscle stem cell sits very close to this, and is vascularized. And so we've been trying to develop strategies to build in the motor neurons to our skeletal muscle culture systems in a dish. And this shows you being able to do that. So here in this movie, you can see that we've been able to, up in the top corner where this cluster is, actually is a cluster of motor neurons that are kind of spreading out through the muscle. And when you do this, either in 2D or in 3D, you can start to see that the muscle begins to contract. And this contraction is a key feature of maturation of the skeletal muscle. It enables us to build a better developmental model or disease model in a dish. And we're trying to develop these in 3D, so skeletal muscle organoids. And we really want to look at how the motor neuron talks to the muscle at these sites. They're called neuromuscular junctions. And where these interactions occur, we think we can model other neuromuscular disorders such as ALS. And so we're trying to build this platform. And again, this support by this endowed chair would enable us to take this in a new direction and think about screening for drugs that would have an effect also on preventing this or slowing the progression even of this devastating neuromuscular wasting disease such as ALS. So our looking forward to future strategies are to think about overcoming the barriers to gene editing delivery. Targeting the muscle stem cells will be needed for a lifelong cure. You can do this by targeting the stem cells directly or replenishing them. So we're doing that, thinking about ways to not have to deliver so much of this gene editing platform to keep it safer is another avenue we're exploring. We're also trying to take our single cell sequencing data and induce maturation to these gold standard muscle stem cells in a dish. We're trying to support them better so that we can use them in a cell-based therapy. And then we're building models to look at neuromuscular diseases in a dish. And finally, something I'm really excited about is I'm also trying to develop a cell and gene therapy class for UCLA undergrads where we want to merge where I want to merge applications of industry and academia. There's so much exciting industry around gene therapy, gene editing. It's exploding into this arena, and I want our young students to see this interface. And so the plan is to kind of have an outside speakers to come speak to these students interested. And I think it's going to be a fun combination of basic and translational and industry learning curve for all of us. So I'm excited about that, which will also benefit from this award.